Now we're going to look at what is healthy for me. So not what makes me overweight, but what kind of foods are healthy or not for me. Of course, being overweight is unhealthy, so that's one part. But even at normal weight, there are healthy and unhealthy foods. And genes do play a very surprising role in this. And so I'm going to start with the concept of the food pyramid that I'm sure you've seen before. So this was a very well-intentioned approach to give one recommendation that should appeal to everyone. It does seem to make sense. As I said, it's well-intentioned. So for example, sweets and animal fats, very unhealthy. So it's a very small piece of my big base of my nutrition. And dairy products is a great source of calcium. So let's do that twice per day. And cereal products has a lot of fiber, so it's good for digestion. So let's eat that several times a day. And red meat is a good source of iron that we need. And so 2 or 3 times a week is a good amount. So it all sounds very sensible. However, geneticists come along and say, hold on, there's an exception. For example, for Caucasians, white people, drinking milk twice a day would be a good source of calcium. However, 20% of them have a genetic defect in the LCT gene that makes them lactose intolerant. And they shouldn't be drinking milk twice a day. It would be highly unhealthy and uncomfortable. So just one gene defect already gave us an exception in the healthy nutrition guideline that should apply to everyone. And let's continue. How about the fiber-containing cereal products? Yes, good idea, but not if you have the genetics for celiac disease and you're actually reacting to gluten, to protein in this. And so suddenly you shouldn't be eating gluten-containing cereal products. And red meat, great source of iron, but not if you have a gene defect that already absorbs too much iron from this food. So you're already absorbing too much due to a gene defect, and we shouldn't increase it more. We should actually reduce the iron intake because your body can't get rid of it, and so you could poison your body essentially. So what you see here, that's one gene, that's 2 genes in one. We have 4 genes and already 3 exceptions from normal nutritional recommendations. And the reality is, in 16 years, we have never found one person where this applied to. Everybody has his own exceptions of how they should be eating. Everybody is different. Everybody is unique. And so that also means we are going to have to follow unique rules for healthy nutrition for everyone. And that's exactly the concept of nutrigenetics that we're focusing on. Now, one very good example is coffee. Now, coffee is a very interesting kind of type of food. And these are all the different genes and gene defects that we're measuring. And as I've explained before, every gene defect has a certain effect, and that needs to be confirmed by at least 3 different studies by 3 different scientists. And if they all independently confirm it, then we know that this is a true effect, or we have a very high likelihood that this is a true effect. So all of this is very well researched. And I'm just going to tell you some highlights. So we're talking about this one gene. I could be talking about all of them, but I'm just telling you the story of the CYP1A2 gene. So coffee, very interesting type of food because there's hardly any other type of food that has a higher concentration of antioxidants, polyphenols, flavonoids, and resveratrol. They're full of super healthy substances. If only there was no caffeine. Now, caffeine, you need to know, was developed by plants to kill off insects. It's a neurotoxin. It poisons insects that eat the plant. So we've realized that getting up in the morning with this neurotoxin makes it a little easier. And so we are taking this toxin, this neurotoxin that kills off insects, and we put it into our body with our morning coffee. Our body knows that this is a toxin, a poison, and that's why we have the protective CYP1A2 gene that comes along and neutralizes caffeine before it could do any harm. And all that remains are all of the healthy substances. So great gene, it detoxifies us from this toxin that we're intentionally adding to our body. Now, you can remember this from virtually all genes. There's some exceptions, but virtually all genes we have 2 copies, one from our father and one from our mother. And so here we have 2 copies. And if both work fine in the way that we would want, then caffeine is broken down, healthy substances remain, and this is a healthy drink. What does science say? There's a study that has shown that if you drink coffee, as opposed to if you didn't, you had 1/3 less heart attacks. So it's heart attack protective. 1/3 fewer heart attacks than if you didn't drink coffee. And so you would say, great, coffee is a healthy drink. Everybody should drink coffee to protect the heart. However, geneticists come along and say, make everything a little more complicated, because these genes don't work in all of us in the way that we would want. Around half of us have less effective or ineffective versions of these genes. So what happens? Caffeine comes in, it's not neutralized fast enough, it remains in the body and does more damage than the healthy substances would help. And so what happens, if only one of the two genes is broken, like that's what I have, that's my personal result, breakdown is only half as fast, and suddenly caffeine does a lot of damage. And suddenly science shows us that drinking coffee doesn't reduce my heart attack risk by a third, it increases it by 2/3. So this gene defect has severe impact on the effect of this one drink that's healthy in other people. And then there are also the people with two defective genes, and they have the same effect. And as I said, around half of the population have this, and the other half have this version. So that's interesting. You can, if you haven't done a DNA test before, you can kind of estimate in what kind of group you could be. It's not 100%, there's some tolerance effect and so on, but typically, if you drink 2 coffees before going to bed, you're typically in this group. If you say, well, after 5 p.m., it's better to not drink a coffee, then you're typically in this group. And I know a person that has this. He says if he drinks coffee in the morning, he's got ringing ears all day long. He can't sleep for a whole night and has ringing ears for another day. So he's very, very sensitive. So these are the 3 different groups that we have. As I said, it's not 100% typically because I personally am on this, but before I knew about this, I drank so much coffee that I had tolerance, so I thought I was this, but unfortunately I'm not. So it's not 100%, so taking out the genes does make sense. But essentially shows the gene makes a difference. Now, the first question that people ask me is how much coffee can I still drink if I have this gene defect? And this is actually something that a study looked at. So here you can see the average population risk for heart attack of non-coffee drinkers. And what you can see, they looked at different amounts of coffee. So with good genes, this happens. So one cup is perfect, a third less heart attack, gets a little less, and then only at 4 more cups, we're back to baseline. So with good genes, coffee is great. With bad genes, like myself, 1 cup is still okay, and it goes up. So if you have the gene defect, 1 cup with caffeine is still okay. So that's around 100 milligrams per day. That should be your limit. And then if you drink more, it severely affects your heart. Of course, as a solution, caffeine-free coffee, because that doesn't contain any of the bad stuff and all of the good stuff. And so that's a combination. Some people mix it. Some people drink caffeine in the morning and then decaffeinate it the rest of the day. So there are different ways of getting around this. But it's important to know that we react differently to this kind of food based on a gene defect. So the next one is folic acid. Folic acid is actually one of the most popular nutritional supplements because doctors agree, even doctors that typically say eat healthy and forget supplements, even those agree that folic acid is important during pregnancy because we found that folic acid deficiency can cause something called spina bifida. It's an open back, so the baby is born with an open back and it's paralyzed, it can't walk. And this is going to be a lifelong effect. So folic acid deficiency is severe, and that's why they recommend 400 micrograms of folic acid for pregnant women. This is of course important for all adults because it's important for methylation, for heart health and so on. So it's not just pregnant women, this applies to all of us. So out of all of these genes, now we're talking about this one, the MTHFR gene. So there's a little story about this. So folic acid is a synthetic nutrient. So something we create in a pharmaceutical manufacturer. And this substance, when it gets into the body, it's quite peculiar because it has no function. It does not have any biological function in our body. But there is the MTHFR gene that has the ability to recognize folic acid and then convert it into the active form called methylfolate. And this is what is healthy for the embryo and for the heart. So we're taking this synthetic inactive nutrient and is activated. Great gene, we need this for folic acid to make any sense. And this conversion works in a third of the population really well because they have good genes. In other people, these genes don't work in the way that they should. And what happens, folic acid gets into the body, it's not activated because the gene is not here and it's not doing its work. And now the tricky thing is, a doctor might say, you're pregnant, this is really important, let's do a blood test and make sure you have enough folic acid in your bloodstream. And the test would say, we found a lot of folic acid, and you would think everything's fine. But the reality is it's inactive, it doesn't work, and so it's useless. So the blood test gives you some insights, but it's not the whole story. So it remains inactive. And this is in 5% of the population. This means every 20th woman that is pregnant is taking folic acid and it doesn't work. So that's a problem. What can we do? We cannot fix this gene today. So what we can do is we can take methylfolate, the active form, to go around this issue. Now, this is, if you don't have genetic information, that's always the right approach. If you have the genetic information, there are two approaches. One of them could be still stick with methylfolate. The other one could be, let's go for folic acid. There are arguments for both sides. So one of them is folic acid, for example, even though it's synthetic, it's absorbed twice as well. So you get more into the body with folic acid. It's also more stable because methylfolate, when you heat it, it's contained in foods. But if you cook that too strongly, then it degrades. So it's less stable, it's much more expensive and less well absorbed. But if you do it in the right way, you supplement it the right way, methylfolate always works, and folic acid only works if you have the right genes. So those two approaches apply. And there are different levels of how quickly you can convert it into the active form. So that's different. So then let's talk a little bit about science, because the fact that this gene defect keeps folic acid inactive and you have deficiency, even though you have you've taken folic acid, this has already been known to us embarrassingly long. So in 2002, there was a meta-study. And that's what we love with scientists, because meta-study means somebody said, there are so many studies on this, I'll do a new one that combines all of that knowledge. And this is a very powerful approach, because then we have so much confirmation. It's very big study size because you can add them all together. So we love this meta-study. It's a very powerful information we can get from this. And in 2002, we already had a meta-study on 24,000 people that already confirmed this. So it was already known in 2002. And then it was done again and again and again. And in total, you will find around 300 studies that show that this gene defect keeps folic acid inactive in any population that has been tested. So this is as well studied as anything in science. And if anybody ever, maybe a doctor or some expert says, nutrigenetics, that's not scientifically valid, you can tell them about this example, because we know more about this than most other aspects in medicine. So this is rock solid science. And then there are even better studies that we love as scientists, and those are placebo-controlled studies. So some people get a placebo, so an empty pill, and others gets the nutrient, and nobody knows. And then you measure what the effect is. So any psychological effects of people interpreting that it helps them are eradicated by this. And also they have shown that people with functioning genes do benefit from folic acid and methylfolate. With the gene defect, they don't benefit from folic acid, only from methylfolate. And there are 11 placebo-controlled studies on this. So very, very good science that has shown that this is a real important effect. That's one of the best studied effects in the field of nutrigenetics. So what you can do is, if the MTHFR gene works, folic acid works, methylfolate, not absolutely necessary to take because your body can convert it and produce it itself. You can still take it, but that's one approach. However, if the MTHFR gene doesn't work, then folic acid is useless. You can skip it. It's just a waste of money. It's not going to do anything for you. You need methylfolate. And so having this genetic information, you can use this to change your supplementation or nutrition. Methylfolate is in certain types of food to counteract this genetic effect that we've found. And just to show how powerful this is, there's one study that has found 12 women that were diagnosed to be infertile, meaning they couldn't get pregnant. They've tried for over a year and they couldn't get pregnant. And these were known to also have this MTHFR gene defect. And in this study, at the beginning, as was expected, they were given 400 micrograms of folic acid. That's standard. But you and I, we know this was useless because they couldn't activate it. That was completely useless, waste of money, had no effect. However, during the study, they said, okay, let's stop this. Let's give you methylfolate, and let's give you more because it's less well absorbed. So they got 2,000 micrograms of methylfolate, and within 1 year, out of 12 women, 11 got pregnant. So that shows the power. This is not a drug or a hormone or some treatment. This is nutrition, a nutritional supplement coupled with genetic information. And this has shown how powerful this information is, just applying the right supplement with genetics. Of course, be careful. Don't think that folic acid cures all infertility. These women were infertile due to the folic acid deficiency. There are other cases where folic acid has no effect on it, but this specific type was solved like this. So this shows nutrigenetic information is extremely powerful if you're using it in the right way. The next one is vitamin D3, a very interesting effect. So vitamin D is one of the most measured nutrients that you would measure in a blood test if you care about nutrition. It's called the sun vitamin, so it's in food, but your body also produces it if you expose your skin to the sunlight. And this time we're talking about this gene here, the VDR gene. Now, the VDR gene sits in the cell nucleus in every cell, and the gene itself has the instruction manual to build a receptor. A receptor is this green grasping arm, and vitamin D can attach to this, and then when it does, the receptor sends a signal to the cell. And so essentially these two have attached vitamin D because that's coursing around the blood. So they have activated it, and these receptors now tell the cell, we have enough vitamin D, do something healthy, in simplified terms. So this activates lots of healthy processes, other genes and so on. So that means for vitamin D to work, we need this receptor coming from this gene to inform the cell for it to actually process and create a benefit. And this is exactly why we do blood tests, because if we don't have enough vitamin D, this doesn't happen, and that's bad. That's a standard deficiency, and so low vitamin D in blood is bad. Now, genetics comes along and makes it a little more complicated again, because in some people, this gene is defective. It has a spelling mistake, and so the instruction manual for this receptor is still there, but it's malformed, so it has a different shape. And so it looks kind of like this. It can still attach to vitamin D, and it can still send a signal, but this attachment, like a lock and key, is so much weaker that it doesn't suffice to inform the cell. So the information, the normal amount of vitamin D in the blood, informs the cell way too little, and it says activate healthy processes, and the cell still doesn't know. So that's a tricky one, because if you have this gene defect, you have normal amounts of vitamin D in your blood. You would think, wow, everything's great. The doctor says, all good. But the reality is your body has a deficiency because it cannot recognize the amounts of vitamin D that you have. Now, interestingly, scientists have tried to figure out what we could do in this case. Because, again, we cannot fix this gene yet. But there's one thing we can do. We can increase more vitamin D. Because if we increase to very high amounts, so the upper healthy range in your blood, not the normal, the upper limit, when you do that, all of the defective or the less effective receptors attached to vitamin D, and in total, they're again able to inform the cell. So that means with a gene defect, you need higher amounts of vitamin D for the same effect than if you had functioning genes. So that shows you need a higher amount of a certain nutrient for the same effect based on your genetics. Again, the blood test just shows you how much is there, but genetics gives you the information how much you need for the effect. And that's one of the interesting aspects of nutrigenetics. So we would check the gene. If it works, normal amount of vitamin D is typically enough. However, if the gene is defective, we need higher amounts of vitamin D for the same effect. Lactose intolerance is an interesting story because it is actually the normal type, and being able to drink milk as an adult is actually the genetic defect. So let me tell you the story about this. So here we're talking about the LCT gene. And this gene has a long history. So around 20,000 years ago, when humans were still in Europe, we were hunters and gatherers, and we were hunting around. We were hunting for animals, anything we could catch. And during that time, every baby was given breast milk, because that was the only food source for a young baby at that time. And breast milk contains lactose, milk sugar. So the mother produces it, and it's an important energy source that the body needs to fuel the body of the baby and the growth of the baby. So lactose is in breast milk. However, lactose is a peculiar type of sugar because it's too big for the body to absorb. It's a huge thing. So it's kind of like two Lego bricks. So that they stick together, but they cannot be absorbed, so the body cannot do anything with it. And that is why we have a gene called the LCT gene that produces a small enzyme. It's kind of like a little cleaving machine that comes along, that's in the intestine, and separates those two sugars into two smaller sugars called glucose and galactose. So they're small enough to absorb, and so the baby can absorb it and benefit from lactose. So that's great. We need this as a digestive enzyme to digest breast milk lactose. So this is what happened in babies. When this baby in the Stone Age grew up, suddenly it didn't have access to milk anymore. So because there was a supermarket, there were no animals, breast milk was gone. So nature said, well, if we're not drinking milk as an adult, why do we need to produce this enzyme for the rest of your life? And so essentially what happened is a switch-off mechanism developed. So the switch-off mechanism said, the older you get, the more we turn off this gene, and this turned-off gene then leads to no more of the enzyme being produced, and that's great because I was just wasted energy anyway, because there's no more lactose, there's no chances this grown man will ever drink milk anymore. And so, great, energy-saving mechanism, all is good in the Stone Age. So the normal type, this is what it looks like. At birth, we produce a lot of this enzyme to be able to digest milk, and then it goes down. For some people it's faster, for some it's slower, but the production of this enzyme goes down, and we lose the ability to digest milk or lactose. And then 7,500 years ago, we changed our lifestyle. We gave up being hunters and gatherers, largely, not all of us, and we began farming. And so suddenly we had our farmland and we had animals that produced milk. And so suddenly, potentially, we humans had the ability to drink milk as an adult. And this is what happened. If one of those early farmers said, okay, the last crop didn't work so well, I'm hungry, we're running out of food, there is a goat or a cow and it produces milk, let me drink that because I'm hungry, what would have happened is the following. The lactose comes into the intestine, the enzyme is off, we're not able to digest it, it stays in this huge form, the body cannot do anything with it, it's useless, and then bacteria and fungi in the intestine say, hey, great, free food, and they start to convert the lactose into gases and acids and cause severe digestive problems. And this is what we today call lactose intolerance. So it's actually the normal type. So instead of us digesting it, it's the microorganisms. And this person very likely stopped drinking milk after this. So this is what happens. So the lower the amount of the enzyme is when you drink milk, the worse your symptoms get. Normal type. Then around 6,000 years ago, in Scandinavia, something interesting happened. In a single person, a genetic defect, a spelling mistake happened in Scandinavia, somewhere around Finland or Sweden. And up here, the following, this gene defect did something interesting. It didn't destroy the gene because that would have been fatal, immediately selected out of the gene pool, but it turned off the up-switching mechanism. So as a baby, no difference, but as an adult, this gene remained on, like in a baby. And suddenly this grown farmer was able to digest lactose, the first guy that could do this. And so he could absorb galactose and glucose and benefit from this. No digestive issues. New superpower unlocked. So instead of this happening, this happened. New farmer type was suddenly able to produce this enzyme throughout all of his life. Now, we know that in the last 6,000 years, lots of famines happened. People starved to death, crops failed and so on. And it is believed, scientists believe, that this person had an advantage. Not just him, but all of his descendants that inherited the same thing. If food got scarce, they could also drink milk and have dairy products, which others could not. And that apparently was such a big advantage that while other people died and starved to death, this new trait spread. And so in Caucasians, in Germany, for example, 80% of people have inherited this genetic defect in this one person from Scandinavia around 6,000 years ago. So they have gotten this new trait, and our body, our nature has adapted to the ability to drink milk as from the supermarket or from animals. So this is actually something new. This happened in our biological history. So in total, 68% of European people have this new phenotype and 31% have the Stone Age type. Why do we know it's in Scandinavia? Because that's where most people can drink milk, and the further you go away from Scandinavia, the more rare it becomes. So for example, Italy, a lot more people have lactose intolerance than normal type, and if you go all the way to Africa, virtually all of them are what we would consider lactose intolerant. So this happened in Europe. It happened again separately in a small community in Africa. But this was the big one, because we Europeans like to colonize the world, and we brought this genetic aspect with us wherever we went. And so this is where the ability to drink milk came from, this one Scandinavian person. The other areas are still what we would consider 100% lactose intolerant. So they're still switching up their nutrients. And typically what you see in these cultures is that dairy products are specifically fermented to reduce the amount of lactose, to reduce this issue, or there are no dairy products in their diet. So this is an interesting aspect. Now, the problem with the lactose intolerance test, you need to know about this. There is a way of measuring how lactose intolerant are you right now. And typically you go to a hospital and they will give you a pint of water with lactose in it, sugar water, and you drink it. And then essentially in a simplified way, they say, well, the worse you feel, the more lactose intolerant you are. There are ways of measuring this. You measure certain gases that come out of your lungs and so on. And so you can measure the amount of lactose intolerance that you have at that time. So that's a tricky thing. If this is what happens during your lifetime, here we'll say you're not lactose intolerant, then you're borderline, and suddenly you're positive. So when you do one of those tests, and you've done one 2 years ago and say it can't be that, that's not true, because it changes over time. So this is one thing. And for some people, it looks like this. And for other people, it looks like this. So they're actually fine until 60, and suddenly they become lactose intolerant. So that means if you have the gene defect, the likelihood that during your lifetime you're going to have issues if you drink milk is around 95%. So a very high likelihood at some point, but it could be in your 60s, 70s, 80s. It could be at the age of 6. So it can be very soon or very late. We don't know that. That's something you would have to measure. But the tendency that you're going to lose your production, and actually your normal type, that's something we can read from your genetics. And so the genetic test will give you this insight, and typically you can continue eating dairy products if you feel fine, but you need to be aware of this, that at some point this is going to be an issue for you. There's no difference if you avoid it, you're not changing anything, so you can really continue eating your dairy products, but look out for it, because typically it takes 5 years of digestive issues before people get the right diagnosis to stop or avoid lactose. So if you have the genetic trait, you know about it, keep eating your dairy products, but look out for it. And as soon as you start developing symptoms, you can stop taking or eating lactose-containing fruit. Good. So the two genes, the two farmer genes, that's 32% of the Caucasian population, they can digest lactose. Interestingly, having one functioning and one that turns off is the Stone Age is the one that turns off. So I've switched it. This is actually the gene defect now, even though it's a normal type. They still have one that works during adulthood. So they can also drink milk. They're just carriers. And only the ones that have the two Stone Age types, they are the ones that have a high likelihood of developing lactose intolerance over the time.